Hi, this is the Biology Paper 2 Foundation, right, for the 2023 series. Okay, so question number 1. Figure 1 shows a place where peat is being removed from a peat bog. Okay, so that looks to me like it's a bit like a cliff there. Okay, and then you can see it all the way along. Right, so what they've done is they've taken loads out, kind of almost like a bit like a cliff. Why is peat removed from peat bogs? To increase biodiversity. A load of rubbish. Biodiversity is a kind of a lot of biological sort of differences within an area. To produce garden compost, yes. Okay, to produce rare habitats, no. 1.2, peat can decay. Name one type of organism that causes decay. All right, so what we've got here is, there's kind of two options here. Microorganisms, and what they will do is they'll do respiration when it decays. So they've got the food, which is then kind of like the peat. They've got warmth and they've got moisture. Well, you could get away with bacteria, okay, because it's mainly bacteria that then do the actual decay itself. 1.3, complete the sentences. Choose answers from the box. Carbon dioxide, nitrogen, oxygen, sulfur dioxide. The organisms that cause decay respire. So the gas used for respiration, right? So respiration, so we're talking about the gases that are used. So we've got oxygen is used for respiration and carbon dioxide is made in respiration. But also obviously don't ever forget oxygen and glucose makes carbon dioxide and water. So the gas used is, in respiration, oxygen. The gas produced by respiration is carbon dioxide. Don't write CO2, even though you're kind of correct, you just never know, right? So make sure you write it out in full like I've done. Okay. Peat bogs cover a total area of 3,700,000 kilometres squared. In 2020, 46% of peat bogs were frozen. How can the area of peat bogs that were frozen be calculated? So what we need to do, we need to do 46% of that. Okay, so 46% of that, I would tend to do it as a fraction like that. We do 46 over 100. So it's the area multiplied by 46 over 100. So it is that one that's the correct answer. 1.5. The percentage of peat bogs that are frozen decreases each year. Suggest why the percentage of peat bogs that are frozen is decreasing. Let's talk about global warming. Okay, so if the whole world is kind of getting warmer every single year, it's gradually creeping up, then that is then the reason why all the peat bogs are then defrosting. You could also possibly there have climate change. Okay, so that is then the reason why they are defrosting. 1.6. Which two human activities decrease the area of land available for other animals and plants? Okay, but two other activities decrease it. Building, okay, recycling plastics, no. Replanting hedgerows, no. Reusing glass bottles, no. Quarrying. So really a lot of the things that we're actually doing, right, as humans to destroy habitats is more building and more quarrying. Okay, you could also have waste disposal, landfill. Okay, that's kind of another option that you could have. Okay, question number 2. A student investigated the fact of either seeing or hearing a stimulus on reaction time. Okay, so what I'm thinking now is I'm thinking ruler drop, all right, but obviously you're going to be a bit difficult to do something to do with hearing with a ruler drop, so we'll have to see what the method is. First, the student measured their reaction time to seeing a colour change. Okay, sit in front of a computer with a reaction timer program going. Press a key on the keyboard as quickly as possible when the computer screen changes colour. Okay, so it goes from white to black, whatever it might be. Record the reaction time. Repeat steps 1 to 3, 4 more times and calculate a mean. So we've got a mean, right, of the reaction time it takes for the screen to change colour. Next, the student measured their reaction time hearing a sound. This is the method. Sit in front of a computer screen, right, with the reaction time program open. Press a key on the keyboard as quickly as possible when the computer produces a sound. Record the reaction time repeat, calculate a mean. Okay, sounds quite good actually. Right, table 1 shows some variables in this investigation. Right, let's just think about it. Don't mess, in class, stay calm. So the dependent is what we are measuring during the actual practical itself. Okay, so we are measuring our reaction time. Independent, don't mess, in class. Independent is what we change between the practicals type of stimulus. So it's either hearing or seeing. Control variable. Okay, God, I've even started filling this in without actually reading the question. Control variable is distractions. So you can't have distractions from the background. Okay, I'm actually going to read the question now, which is completely and utterly stupid. Identify each variable as an independent variable, a dependent or control. Tick one box in each row. Job done. 2.2. How could the method be improved? Right, so let's think about it. Method improved. Measure the reaction time with a stop clock. Not a chance, okay? Because you're using a computer program that will detect when the key gets pressed. Only test reaction times to seeing a colour change. Well, what's the point in that? You've got nothing to compare it against. Repeat both 10 times. The more times you repeat, the better it's always going to be. 2.3. A shorter reaction times means the student reacted faster. The student reacted faster as each test was repeated. Right, okay. They're getting better at it. Suggest one reason why the students' reactions got faster. That could be practice improves performance. Okay, that's obvious, isn't it? The more time you play sports, the more times you kick a ball, the better you get at it. Okay. Fewer distractions as you kind of get used to doing it. And it could also be familiarity with the test. Okay, the first time you do it, think, oh, I've done it right. And then what you do is you then do it more and more. Table 2 shows the results. Seeing the stimulus and hearing the stimulus. So seeing the stimulus, right, the mean reaction time in milliseconds was 350. Hearing the stimulus was 220. So it sounds to me like seeing the stimulus, right, was slower than hearing the stimulus. So you reacted faster to hearing it. Right, 2.4. Okay, so what I've got to do is I've got to do this information. Plot the data from table 2 on a bar chart. Okay, so what I'm going to do here is, this is going to be difficult for me to do. That's going to be seeing, so that is that column there. And then what I'm going to do is I'm going to move along and I'm going to do that one there. Okay, now I could do that, but obviously I'm only going 2 bars on it. So what I'm actually going to do is I'm actually going to rub that out. Okay, and then I'm going to do that one and that one. And then I'm going to do that one and that one. So that is seeing. And that is hearing. Okay, then what I've got is I've got my results. Okay, so for seeing it goes up to 350. Make sure you use a ruler. Now I obviously can't use a ruler on this, and it's pretty obvious I'm not using a ruler. There's my bar. Hearing was then, I think it's 220. Okay, I've just got it written on a piece of paper. 220 goes up to 220 and across. Now what you've got to do is you've got to make sure your bars are the same width. You use a ruler like I haven't done. It then says plot the data on the table as a bar chart. I've just done that. Label each bar, seeing, hearing. Label the y-axis. So this is then my y-axis. So this is then mean reaction time. Okay, 2 marks. 2.5. Compare the reaction time when seeing the stimulus with the reaction time when hearing the stimulus. So the seeing reaction time is longer than the hearing reaction time. Can't be lazy there. Now what you could obviously do, do it the other way around as well. The hearing reaction time is shorter than the seeing reaction time. You could do it vice versa. Question number 3. Okay, so what I've got on here is a student investigated how the distance from a river affects the number of different plant species. So there's our river. I'm going to colour in blue because that's a nice blue colour. Okay, shows the river between 2 fields. Okay, field number 1, field number 2. 3.1. A, B and C show the position of 3 transects. So that's then your tape measure that goes from the river into the field, and it could be 30 metres long. Where should the student position another transect for this investigation? Right, so what I could do is I could do one there, as long as it's right angles and the same length. Okay, that should be right angles. I could do right angles to the same length. I could do another right angle there. Okay, it's anything as long as it is right angles to the actual edge of the river. 3.2. The student recorded the number of different plant species at 1 metre intervals along the transect. Okay. Which piece of equipment should be used at 1 metre intervals along the transect? A microscope, that would be pretty cool, wouldn't it? Looking at the actual individual kind of insects through a microscope. It's definitely going to be a quadrat. Your quadrat, your square, could be 1 metre by 1 metre, or you could have another one. Sorry, that's very badly drawn. 50 centimetres by 50 centimetres. Okay, that should be a square. Right. What was the mean number of plant species 5 metres from the river? Right. So the mean is our little cross. And then there's obviously a bit of variation, kind of uncertainty, and it goes up to there, that's the maximum, and there, that's the minimum. So let's have a look at the question again. What was the mean number of different plant species 5 metres from the river? 5 metres. The mean is there, and if I go all the way across, 6. Question number 3.4. What is the effect of increasing distance from the river on the mean number of plant species? Okay, so the mean number of plant species, because on your graph, right, I haven't got it on the page here, it kind of goes up in that direction, doesn't it? So the mean number of plant species, mean number of plant species increases away from the river. Okay, now I could have put increases, right, but you've really got to be dead precise. You've got to treat the examiner as being completely utterly thick, right? You've got to say absolutely precisely what you actually mean in an answer. Question 3.5. The minimum and maximum values on figure 4 show the range of results at each distance. Okay, so what we've got is you've got your cross in the middle, and then you've got a line there like that, and you've got a line there like that, where the cross in the middle is the mean, that's the max, and that's the minimum. Why is it useful to know the range of results? Okay, calculate a mean, no, you got the mean. To know the uncertainty, now the uncertainty is the distance on either side of the mean. Question 3.6. Cows are kept in fields. That's fantastic. That's the biggest thing I've learned all day. Fewer plants are found where cows often walk on the ground. Fair enough. The number of cows is a biotic. Biotic means living. Okay. Which two factors are abiotic? Okay. First time I read that, I got it wrong, and I thought, oh, it's biotic, and I looked down there for biotic things, right? But abiotic, right? So consumers, no. Light intensity will have an impact, and that is an abiotic factor, just like moisture. That's biotic, that's also biotic. So those are then your 2 answers for that one. 3.7. Explain why a decrease in the number of plants across the world increases global warming. So decreased plants, world, increases global warming. Right, so we've got to think now that as we decrease plants across the world, why does global warming increase? Okay, So there is more CO2 in the air. So there is more greenhouse gases. Making, okay, I'm going to be really lazy here, global warming. Okay, and it could also be due to less photosynthesis. Okay, so now make sure, explain, chucking a so. Okay, so's good. For question number 4. Okay, salmon are fish. Well, this is quite an educational paper this time. Salmon are fish. A species of salmon has the binomial name Oncorhynchus. Okay. 4.1. Table 3 shows the classification for this species of salmon. Choose the answers from the box. Right, so I've got an empty box, empty box, empty box. Okay, now the way that I always remember this, whether it's right or not, is King Philip can order 5 giant sausages. Okay, now there's loads of different ones that you can do for that, little poem for it. So kingdom, phylum, class, order, family, genus, species. Okay, so kingdom. So they are an animal. King Philip, phylum, can order 5, so they've missed out the can and the order, class and order, family, genus, species. Genus, species. Okay. What we got here? Figure 5 shows sea lice attached to the skin of the salmon. That looks pretty mean. Sea lice are small animals that feed on the skin and blood of salmon. On a salmon farm, the salmon are fed with food made from soya. So salmon have soya. That's their food. Okay, I guess that they eat it. Right. Which food chain represents the salmon, sea lice and soya? So soya starts, salmon, sea lice. That's supposed to be a tick. Very bad tick. 4.3. What do the arrows on a food chain represent? Different numbers? No, it's nothing to do with numbers. Predators? No. Producers? No. Transfer of energy? It is the transfer of energy. Okay, and sometimes I might even think about it. If you've got soya and then you've got salmon, sometimes I think about it as eaten by, right? But it is actually the transfer of energy. So the energy in the soya gets transferred to the energy in the salmon. 4.4. Some salmon are more resistant than other salmon to sea lice infections. That's good. Salmon farmers selectively breed salmon. Okay, so what I can do is I can think they're already trying to get rid of the salmon that are more susceptible to sea lice. Salmon farmers select salmon that are the most resistant to sea lice and breed these salmon together. Fair enough. What is the next stage in selectively breeding salmon that are resistant to sea lice? Okay, so what we've got here is breed together the offspring that are most resistant to sea lice. Well, of course. So we'll look at the next. Killing the offspring that are resistant to sea lice. Well, why would you do that? Remove the genes for resistance to sea lice from a selected salmon. That sounds incredibly difficult. 4.5. When is the process of selective breeding finished? After one generation, not a chance. When all offspring are resistant to sea lice? Yes. When sea lice are living on salmon. On all salmon. What do you want them all salmon for? Right? It's when all offspring are resistant. So what you've done is you bred it out of the salmon. 4.6. Salmon that do not have sea lice are more profitable for the salmon farmer. Suggest one reason why. Well, I assume salmon are going to be larger. They are going to grow faster. okay, because they haven't got sea lice that are then eating their food that's inside them. It's not off-putting, because I have to admit, if I bought a salmon from Morrison's, right, or wherever it was, and it had like little lumps and bumps on it from sea lice, I'd be a little bit dubious about it. So there is less skin damage. Okay, you just got to think about what would it look like. 4.7. What is a disadvantage of selectively breeding salmon? Okay, all the salmon may suffer from the same disease, correct, right? Because if you selectively breed anything, what you're doing is reducing the amount of genes in the population. Fewer sea lice will infect the salmon? No. The salmon will have a large variety of genes? No. 4.8. Okay, other fish farmers have produced genetically modified salmon. Genetically modified salmon grow large enough to sell in 18 months. Non-GM, 3 years. Sounds good, 18 months. GM salmon need 25% less food to get to the same size. Things are looking good. Suggest 2 advantages of farming GM salmon instead of farming non-GM. More profit. Okay, that's what it's all about at the end of the day, isn't it? There is less feed required. They grow faster. Okay, and it's not farmed as long. Okay, so 4.9. GM salmon are often farmed a long distance from wild non-salmon live. What is the advantage of farming GM salmon a long distance from where they live? Okay, to give GM salmon different conditions. No, they probably want the same conditions. To increase the genetic differences between GM salmon? No, because we're kind of genetically informing them ourselves anyway. To reduce the risk of GM salmon? Correct. Okay, because we don't really want them breeding together, because if we've got wild salmon, we want to keep wild salmon as they are. Question number 5. Homeostasis is the control of internal body conditions. Control of body temperature is an example of homeostasis. So homeostasis is basically keeping everything the same internally in your body so that the enzymes can work at their best, at their optimum. Draw one line from each part involved in temperature control to the function of its part. So the brain changes air temperature outside of the body. Why? Brain contracts to increase body temperature. No. Coordinates information. You get your ruler out and you draw a nice straight line in between. Muscle changes air temperature outside the body. Don't understand. Muscle contracts. Got to be a muscle because muscles contract. Receptor changes air. Why? Detects change. So that's then detecting the change. Okay, it's process of elimination, that question, really. 5.2. Why is homeostasis important? To allow cells to function properly. Yes, that's what it's all about. To change body temperature, so you want to have, you're in a room at 15 degrees C and you want your body at 15 degrees C? No. To decrease water levels in the body throughout the day? No. It actually makes you feel ill. To maintain the optimum conditions for enzymes? That's the best answer of them all. To prevent reactions inside cells? No. What graph are that graphs? The body temperature of a long distance swimmer can change with the length of time swimming in cold water. Fair enough. Okay, so what we've got here, so body temperature at the start, 37. After 4 hours, 35.5. Figure 6 shows how the body temperature of one swimmer changed in the first 4 hours of a 6-hour swim. Describe the trend. Okay, so what we're doing is then, we're just kind of looking for the pattern, right? And I'm looking at is, as the time increases, the body temperature decreases. Okay, so as the time increases, the body temperature decreases. 5.4. Determine the change in body temperature in the first 4 hours of the swim. All right, well, I've already did that even without thinking about it. So we had 37 degrees to start off with. It went down to 35.5 degrees. So that is then 1.5 degrees C. Determine the change in body temperature. So the change in body temperature is 1.5 degrees C. Okay, I got that from the graph. Hypothermia is a dangerously low body temperature of 35 degrees or colder. Okay, we haven't got anywhere near that yet. Well, I suppose 35.5 is not far off. Predict when the swimmer was first at risk of hypothermia. So we want to know is when they are first at risk of getting to 35 degrees. Extend the line. Oh, nice, it's going to be pretty tricky on here. So what I'm going to do is, it looks like a curve. Okay, so what I've tried to do is I've tried to follow the smoothness of the curve. Okay. Then what we've got to do is we've got to look at when do we get to 35 degrees. Right, there we go. There. Okay, so what I'm going to do is I'm going to say 4.6 hours. Okay, which is pretty much where it is on there. Swimming in cold water decreases the insulin concentration of the blood, right? So insulin, what does that do? That decreases glucose in your body, mainly in the pancreas. Complete the sentence, choose the answer from the box. Insulin is produced by the pancreas. Okay. Insulin is a hormone. Which organ system produces hormones? Glands. Okay. Hold on a second. Right, I'm actually doing that. Which organ system? Mr. Leek's a muppet. Endocrine. Endocrine system, right? It's an organ system. I didn't even read it right. 5.8. Complete the sentence. Choose the answer from the box. When insulin concentration in the blood increases, blood glucose concentration will decrease. Don't circle it in there. You've got to make sure you write it in here. 5.9. Give one risk factor for type 2 diabetes. So type 1 is you are born with it. Type 2 is to do with your lifestyle. So it could be obesity, it could be overweight, which I guess is very similar, okay, but that kind of works as well. Lack of exercise, smoking, loads, right? Basically, if you have a really, really kind of bad health. BMI that's too high or even too low. And inactivity. They're just a few examples, but basically it's you live in a very, very unhealthy lifestyle that could bring on type 2 diabetes. Right, question number 1. Sexual reproduction in humans involves the production of eggs, egg cells and sperm cells. Name the type of cell division that produces egg cells and sperm cells. Okay, well, there's only two types of cell division. There's mitosis and meiosis. So MEI OSIS. Now it's one of those words that you actually have to spell right, and if you don't spell it right, then you actually don't get the marks for it. So be absolutely precise on that one. Sexual reproduction produces offspring that are genetically different from each other. Okay, so genetically different. Good. Give 2 reasons why sexual reproduction causes variation in the offspring. Okay, so what you've got is you've got the gametes, so that's your eggs and the sperms, are not identical. Okay, and you're getting genetic information from both parents. Okay, so your egg and your sperm, can't spell parents, your egg and your sperm, right, and then joining it together, right, and then what you're doing is you're going to get genes from mum, getting genes from dad. Okay. Polydactyly is an inherited disorder, right? The allele for polydactyly is dominant, right? Dominant. We've got to remember polydactyly is dominant. A person with 2 copies of the allele little d will not have polydactyly. A person with the genotype big D is homozygous, because homozygous means there's 2 of the same. What word describes the genotype big D little d? That is het. heterozygous. Okay, so this one, if you add little d, that would be homozygous recessive, and it's big D, that's homozygous dominant. 1.4, a person with genotype big D, and a person with genotype little d plan to have a child. Determine the probability that the child will have polydactyly. Complete the Punnett square, so I've got to do that. Identify only offspring genotype that would have polydactyly. So I need to come back to that question a little bit. So what I'm going to do is I'm going to do the polydactyly at the top, and then, oh God, I'm doing it wrong, and then I've got little d. Then what you do is you cross over. Don't know why I changed the colours. So big D, little d. Then I've got big D, so I'm doing there. Little d. Now let me just go back to the question again. Complete the Punnett square diagram. Done that. Identify any offspring genotype that would have polydactyly. Okay, now I'm not quite sure what it means by identify. Okay, but what I'm going to do is I'm going to say polydactyl, and I'm going to say that one has and that one has. So that has and that has. Okay, so I need to identify any offspring genotype that would have polydactyly. Okay, they're the ones. Probability that a child will have polydactyly, 2 out of 4 is 50%. Okay, so we're always doing it as like a percentage. Okay, so embryos can be screened for the alleles that cause inherited disorders. So that's looking for then the genes that are then in there. So what you could have is you could have an embryo, right? And then what you could do is you could check out what genes are in there, what alleles are in there, to see if it is a possibility that they're going to have the disorder. Give 2 advantages of embryo screening. So an advantage could be, well, we'll talk about advantages in a minute, but if you think disadvantage, a disadvantage could be the fact that it costs a lot of money. And also it is kind of playing God. It's looking at the actual genes in the embryo before the baby is actually born. But 2 advantages. First thing is we could find out if the embryo has the disorder. Okay, so the first stage is we could actually find out if it has got it or not. The second one could be prepare for a baby with the disorder. Okay, so if you know that your baby is going to have an extra finger, then what you could do is you can kind of mentally prepare the fact that your baby's going to have an extra finger. If you've got a baby that's going to get cystic fibrosis or potentially get cystic fibrosis, then that's much more serious because that's a much more kind of long-term illness. So you need to be able to prepare and get mentally, I guess, ready for it. You also need to decide if you are having the baby. Okay, so whether that's an advantage or not, right? You have then, if you find out it's got it, then what you need to do, you then have to make a decision about whether you're going to keep it or not. But one advantage is, if you then end up not having the baby with the disorder, there will be fewer with the disorder long term. Okay, because what's happening is the genes in the gene pool for that particular disorder are then decreasing. Question number 2. It is estimated that 99.9% of all species that have ever existed are now extinct. Okay, why is the percentage of species that are extinct only an estimate? Okay, all individuals of one species have the same genes. Well, that's a load of rubbish. Extinction is always caused by humans when it isn't, because the dinosaurs weren't. Humans have not found evidence of every species. Okay, we don't know. There might be some tiny little mouse, right, from 1 billion years ago, right, that we're not aware of. So, you know, we can't say that that actually existed or not. So it is just an estimate. What evidence is used to study species that have become extinct, right? So what we're doing is when we're looking at extinction, okay, and there's only really one piece of evidence, is fossils. Okay, there's no YouTube back then and, you know, all those different things. So it's fossils out of rocks. And that gives us an idea about what animals were around at the time. 2.3. A bacterium called Clostridioides difficile, C. difficile, that'll do, right, can infect the human digestive system. C. difficile can multiply and produce toxins, poisons, okay? The toxins cause diarrhea, nice. Doctors are concerned that new strains of C. difficile may evolve. Antibiotics may not be able to kill these new strains, okay? Antibiotic immunity. Okay. Explain how the evolution of antibiotic-resistant C. difficile can be slowed down. Okay, so it's not asking what antibiotic resistance is, right? It's asking how can we actually slow down, because they're kind of saying, well, it's going to happen, right? How can we slow it down so it doesn't happen as quickly? All right, so we need fewer prescriptions of antibiotics, so less exposure. Okay, so I'll see this here. Explain. What word am I using? So. Okay, or I could be using because, but so works quite well. If you've got it, do not treat mild, mild infections with antibiotics, so the body can deal with it. Okay, that kind of makes sense really, doesn't it? Because then what you're doing is then it's not getting exposed to it. Don't use antibiotics. Okay, I'm just going to write AB on virus, as antibiotics don't affect viruses at all. Always complete a course of antibiotics so all bacteria are killed. Because if there's a couple left, right, then they've got a pretty good chance of actually being immune to it. Right, but the main part of this is it's an explain question. And what we're doing is we're saying so, I put as, so. Okay, so what it's doing, it's coming up with a fact, and then you're then saying so this is then happening. And we're talking about slowing it all down.